Welcome to Chuck Karan, who is got his PhD from the University of Rochester and performed his postdoc at Harvard University. He is an associate research scientist here at Columbia in the Department of Systems Bio, and he is the scientific director of our high throughput screening facility, which is part of the Columbia Genome Center. And so thanks very much, Chuck, and I welcome you to tell us about the new tools for discovery in the high throughput screening facility.
Great, can everybody hear me and see?
Yes, yes, perfect. Thank you.
All right, great, thanks everybody for joining. As Emer said, I'm going to introduce you to the high throughput screening facility and walk you through some of the tools that we have available for people, and also discuss with you one project that we found really interesting and allowed us to develop a lot of new tools that we think the community can use. So just some of the basics. Obviously, you know, our mission, the reason we exist is to help people to develop and implement novel high throughput assays. You know, we're one of those core facilities that sort of is many things to many different people. You know, obviously, assay design and development is our specialty. Data analysis from any resulting screens, developing secondary assays, implementing them. We do have access to different genomic reagents that can either be cherry-picked or screened. Automated high content imaging tools, but at our base, we have access to high quality automated liquid handling. So, you know, in this slide, I'm just pointing out some of the really simplified view of high throughput screening, right? I mean, the major break in high throughput screening is between biochemical assays and cell-based assays. And in general, biochemical assays are considered to be slightly more simple, right? And that's where you have a purified protein and you're really just looking for a small molecule that can somehow inhibit the functionality of that protein. And obviously with a cell-based assay, there's a myriad of different assay types that can be developed with a cell-based assay. So complexity can get rather high with cell-based assays. These are our three main liquid handlers. I'm only going to really, for this moment, I'll talk about the Cell Explorer, which is our main automation system. It is a very traditionally designed high throughput instrument with a center robotic arm and devices arrayed around that center. It has automated tissue culture, automated compound storage, and really provides as close to a sort of a hands-free high throughput assay environment as we have. I'll talk about the STAR and the LabSite Echo later. And then for detection, our main plate reader is the Envision, and that's integrated into the Cell Explorer, but we also have a TCAN plate reader that's offline. For high content imaging, we currently have a GE Cell 2000. So one thing I wanted to point out, because it's probably not apparent, is that we also, beyond sort of the really high-end automation, we have a self-use model that many different labs use, and it's really handy for people that have, you know, a limited number of drugs. You know, for sanity's sake, I would say you probably want to only deal with less than, you know, 15 or so drugs, and you know, you just want to generate dose response curves and maybe some different cell lines or some simple drug combination experiments. We have multiple instruments. I have here pictured the HP digital dispensing system and that TCAN plate reader that we can train somebody in like 20 minutes on how to use it. It's really a straightforward instrument. It's great because it's a non-contact dispensing unit, which means you don't have to worry about compounds sticking to tips, which most of them do. And there's also a minimum dead volume that's needed, so you won't waste a lot of compound using the system. The only caveat to the system is that it's really biased towards DMSO solvated small molecules. There are some aqueous molecules that can be used, but you need to have a rather high surfactant load in order to use the HP. So at the heart of any high throughput screening facility are their chemical libraries or the screening deck. In general, we have two broad classes of small molecule libraries available to users. The first is our bioactive and known drug collections, and that's about 7,000 compounds. All of these compounds have some defined mechanism of action. Whether it's the right mechanism, I can't tell you, but there is a defined target for most of these drugs, whereas our diversity library is about 280,000 compounds, and that is just that. It's a very diverse selection of small molecules. We also have numerous functional genomics tools that are available either for use in high-throughput screens in-house, or if you want individual clones, we can do cherry picks and work with you on that. Okay, so last week's talk by Eris was the perfect segue into my talk today, because then I don't have to talk about algorithms at all, something for which I am not qualified to talk about. So we were charged with making the front end of the precision oncology pipeline that Andrea was trying to develop. And very briefly, Andrea builds these regulatory models of diseases. And what he wants to do is to take different small molecules, perturb cells with them, sequence them, and develop a roadmap for these small molecules and how those small molecules fit into the regulatory model. So you start off with your disease, you look for master regulators and the targets of those master regulators. You bring in your drugs, you perturb the cells, and what you're looking for are small molecules which can reverse the signals of the master regulators. This whole model presented some really unique challenges for us. And so the first thing we had to do is to identify a library that we can use in this project. Now, obviously, as part of precision oncology, this was something that we needed to have clinically relevant compounds. So we built by hand a library, first of 121 FDA-approved compounds. These are frontline treatments in oncology, and every one of those drugs is sequenced every time we do a new disease in cell line. Below that, we have 205 compounds in what I call our experimental oncology set. This is made up of drugs which are in later stages of clinical trials, phase 2 and phase 3, with an oncology inclination. So, and again, our goal is for each one of those compounds to be sequenced in every disease in every cell. And then below that, we sort of have a fill in the blank of bioactive compounds. Now, these are not clinically relevant per se, but could represent unique molecular mechanisms in the cell. And the great thing about this library is that we developed it for one purpose, but these 300 or so drugs are actually used by other researchers for non-precision oncology projects. So these are available to anybody to use as sort of a first pass in clinically relevant oncology drugs. So for the next two problems, I'm going to combine them because really there was one solution for both problems. So if you think about what we really want to do, we have a whole bunch of drugs and we have a whole bunch of cell lines. And we want to treat those cell lines with each drug at a very specific concentration. And that concentration has to be such that we're actually doing something, but we're not killing the cells, because dead cells don't sequence very well and don't give very informative data. So the traditional paradigm for the dosing especially is the hardest part. So the first part, it's just simple. We're going to have to do dose response curves on every drug versus every cell. The second part is once we've done those dose response curves, we're then going to be in a situation where we're going to have to create a plate of small molecules to treat these cells where every drug is at a unique and different concentration. And the traditional paradigm for that would be that we would create a stock plate for every cell line that would be, 10x, 100x at the concentration that we want to deliver into the cells. Doing that on the scale that we wanted to do this project was really untenable. And we would have spent more time creating that plate than we would have done anything else in the world. The other problem too is that's extremely wasteful. You just burn through drugs left and right. But luckily, there was on the market a product that can actually do both of those things. And so we invested in the LabSide Echo, which is an acoustic dispensing system. And so it is exactly what it sounds like. It uses sound to dispense liquid. It has this little, almost like a little speaker, this transducer that sits on the bottom of your source plate, emits sound, which ejects a very regular droplet. In our case, it's a 2.5 nanoliter droplet, which is then, for all intents and purposes, caught by a destination plate, which is actually inverted over the top of the source plate. And so by doing this, you can have a source plate of your drugs, and you can, in that assay plate, do something like a dose response curve without having to have multiple source plates. And also, and most advantageous for us, is that we can have this library of source compounds and then dispense each one of them at a unique and different concentration to the destination plate. So that's great. So we have a solution for how we can automate this process, how we can, you know, deliver these drugs accurately and rapidly. But there was a problem, right? So if you think about what we're trying to accomplish, right? We have 500 or so different drugs that we want to treat versus lots of different cell lines, and then do a sequencing experiment. So the traditional model for that would be, you know, one compound treatment equals one library equals one sequencing experiment. And obviously for this number of samples, this was really an untenable approach. Not only the cost, but the QC that would be involved and all of the individual steps just leading to sequencing, we would never be able to accomplish the goals that we had in that manner. But we were very lucky that Peter Sims, in collaboration with Andrea Califano, had developed this technology called PlateSeq. And it's just that, it's a plate-based sequencing technology. We start with our compound treated cells, lyse them, capture mRNA, and then we can barcode and amplify, pool those samples. And again, here we have the oldest instrument in the facility, the Hamilton liquid handler, which we retrofitted to do all of these really complex liquid handling steps. And then at the end of the day, you only have a single library to generate from the pooled samples. And you know, this has all been published before, but you know, obviously you're not going to detect the same number of genes, but we were really happy that a multidimensional analysis showed that the PlateSeq drug treatments overlapped almost perfectly with the TrueSeq sample. So with that, we had a system where we could really rapidly, you know, test small molecules, treat them, and then sequence them. Great. The next problem is the logistics. So this is an extremely complex project, right? There's data that's going back and forth between multiple different labs. There's information that is needed for each step. So we generate metadata. That metadata has to be shared with the sequencing group, which has to be shared with the Califano lab so that they can process the data. And so what we did is we've created a system of communication back and forth where we start out by doing all of the primary dose response experiments, generate the PlateSeq libraries. Those go to sequencing. That data is then captured by the Califano lab, where they can process and back up the data, send us QC reports, where we created a web portal that now contains all of the metadata for all of those plates. And then from that, drug reports can be generated based on the activity of the individual compounds. And so here I have a snapshot of our end of that web portal, which is the publicly facing end of the portal, where we have information on cell lines, what's been sequenced, and then embedded in there is the metadata for all of those files, so that each one can be processed. Now, all of those tools together led to all of our work in precision oncology, but each of those tools individually can be used and are used by different laboratories to sort of fit whatever purpose they have for them. And it doesn't have to be because they want to do sequencing. Well, obviously, if you're doing plate seq, you're going to do that. But the echo is basically, honestly, at this point, is indispensable and is used in almost every project we have now, just because we can do a primary screen, get hits, and then really rapidly follow up those hits with dose response curves and really stratify the population of hits and make better decisions going forward on what to follow up. So sitting somewhere in a warehouse in Boston, we partnered or worked with High-Res Biosciences to develop a 384-well variant of that plate experiment. So what I showed all along has been done in 96-well plate. So with the 384-well plate seq, obviously we'll be quadrupling the number of compounds that we can screen in each of these experiments. Unfortunately, this is a victim of the pandemic outbreak, and it was supposed to be delivered the week that we shut down. So CRISPR screen. So we, with our automation and the tools that we have, we are, I think, ideally suited to do arrayed CRISPR screens, right? I mean, I think it's a very logical extension of a lot of the work that we're doing. And so that's actually, we're actually working now on integrating some of those CRISPR screens, maybe even into our plate seq tools. So there's a lot of movement on CRISPR, obviously. And lastly, so the one thing that changes very rapidly is always the models, right? And we are a model-driven core. High-throughput screening is whether you're doing a cell-based assay or whatever it is, it's really dependent on the quality of your model, right? The quality of your model is going to really dictate the quality of your results. And more of these models are moving to more complex tissue culture systems. And so, we really want to try to stay on the forefront of that and start to develop some of these confocal high throughput systems. So we've had sort of a really interesting role in Columbia's response to COVID. When I was first consulted by pathology, when they had their serological ELISAs, the idea was that we would use, the Cell Explorer, our big automation systems, to automate and really sort of power through a lot of these ELISA assays. Now, unfortunately, due to the regulatory environment, and we're not a CLIA certified lab, that just never really panned out. And instead, what they've done is they've enlisted teams of manual pipetters to do all of these ELISAs. And so I then was asked to train the human robots on how to effectively use multi-channel pipettes, and also we wanted to have some at least cursory analysis to show that these people were proficient at using multi-channel pipettes. And so I continued to have conversations with pathology. They are buying some of their own automation to do some of this stuff. So whenever possible, I've been consulting on different types of automation and how to implement it. And recently, they started to receive some other commercial kits that have been available on the market. And our plan now is to acquire some controls from those guys and do some side-by-side comparisons of the different commercial kits that are available. In addition, we have our first set of purified proteins. So we're really ready to start in earnest some high-throughput screens on COVID-relevant targets. We've also received some specialized libraries from these are not available for everybody, but for certain projects that were released by different pharmaceutical companies. So we've been prepping those libraries and getting them ready for the individual researchers' research projects. And so that's it. So all of the work done in the high-throughput screening facility is done by my two PhDs, Hailey and Sergey. Ron, who basically runs the place, and Antonina is my technician. If you have any questions, I'm happy to answer.
Thanks so much, Chuck. That's great. I can ask very briefly, how many humans did you have to train?
So yeah, we had about 20 or 22 different people come through.
Okay.
You know, they were all postdocs or staff, and they knew what they were doing. And it was a lot of it was, you know, just so that we would have a record saying, hey, look, they were trained. They, you know, I created basically a qualifying exam in multi-channel pipetting, which may be a first. Yeah, it's okay.
I'd be afraid I'd fail it, but
you know, I didn't want to say it, but I did have to shame some people, but it's okay. They got over it eventually.
And maybe while folks are thinking, please raise your hand or unmute yourself or drop a question in the chat box. And then in terms of, you know, at this time, if people want to reach out to you guys and chat with you guys about planning experiments and projects, once we ramp up again, maybe just say a few words about how available you guys are for that type of consultation.
Yeah, so that's basically been the other half of my life. There's a lot of grants going out, it turns out, even during a pandemic. So yeah, please contact me. My email is on the screen, CK2389. The earlier you tell me that you want to do something, I think the better off for everybody will be. You know, we're happy to help at every step. So.
Great. Okay. Does anybody have any questions for Chuck or for Remy? Please unmute yourself if you want to just speak up, or drop me a question in the chat box. While we're just hanging out here, Chuck, I just have an end slide to share. So if you'd unshare, I can drop that up. We had a great turnout. I really appreciate everybody coming out. And listening. I think we have some great examples here of our shared resources and what is available to all of our researchers. And so we really appreciate everybody coming out. I have to try and share my screen now, and it's not working very well. Okay, for some reason that doesn't want to share. So does anybody have any other questions that they want to ask our speakers? Anything else that you guys would like to raise? In that case, I would like to, again, thank our speakers for their time today. I would like to remind everybody that we do this. Honestly, I'm going to share you my screen. Here we go. We do this every Tuesday and Wednesday. We have tomorrow, we have an exciting interdisciplinary seminar where we're bringing together a basic and clinical researcher to talk to us about translational research in T-cell lymphomas. Larissa Geskin and Teresa Palomero will present an interdisciplinary research seminar, so please join us for that tomorrow at 4 to 5. And then next Tuesday, we are going to take a pause on our Cancer Center shared resources, and we're going to hear from two of our leadership here in the university who have been integrally involved in the launch and the operation of our new Columbia University COVID-19 Biobank. And given all of the different funding opportunities that are currently existing for COVID research, and most recently COVID, or quite recently COVID research in cancer and in cancer biology and cancer patients, we think this will be a very nice resource for your research to hear about. So please join us for that. Also 4 to 5 next Tuesday. Okay, thank you so much, everyone. Thank you very much to our speakers. Really appreciate your time and coming and telling our community, our research community, about the wonderful shared resources that you run. And we appreciate your time, and thank you everybody for joining us. Take care.
Take it easy.
Bye-bye.